Good morning, everyone. I'm Jason, and today my topic is about diet and the mitochondrial DNA haplotype of a sperm whale found that of Jurong Island, Singapore. And we know the sperm whale is the two-turn sperm and whale, but the sperm whale is don't like the sperm. Instead, he have a very big forehead. So why do we call them sperm whales? Whale hunters mistook the spermacetic organ for sperm because they have a lot of white liquid. And the sperm whale's scientific name is Physeter macrocephalus, and their distribution are in the temperate and the tropical ocean. In this map is the color area. And they mainly eat cephalopods and pelagic and benthic fishes and crustaceans and tunicates in small proportion. And the main role in this study is the David sperm whale in Singapore. And she have a nickname Jubilee. And she's an adult and a female whale. And we found her in 2015. And this whale carcass provides us an opportunity to study on her natural history. And the objective of this study, first, we want to determine the diet of the sperm whale from sperm content. And we will describe the relative proportion of each prey specimen and the size of cephalopods. And third, determine the mitochondrial DNA country-region haplotype of this sperm whale. Fourth, investigate the possible location of the sperm whale's origin by using a coastal hydrodynamic model. And what is haplotype? A haplotype is a group of alleles in an organism that are inherited together from a single parent. So basically, we can identify patterns of a genetic variation by examining haplotypes. And the material and the methods, we will collect approximately 80% stomach contents. And the mass is by morphological identification. And we will do pre-sorting for the upper and the lower cephalopod beaks. And after we get the beaks, we measure the lower rostral length to calculate the dorsal length and the mass by the conversion formulas. And they make us quantify the cephalopod trace. And mitochondrial DNA haplotype, we collect a sample of skin and skeletal muscle, and a section of the mitochondrial DNA country region was targeted using primers and PCR protocol from this study. And the country region is a part of mitochondrial DNA, and the circle is mitochondrial DNA. And we got a result sequence we aligned and added using the software sequencer and the haplotype matching followed by this paper. And the coastal hydrodynamic model are useful for investigate flow circulation. And we choose the model from this paper as the hydrodynamic model because this model has a good representative of flow in the Singapore Strait and the surrounding region. And in order to serve a substitute for GB, drugs were released during the simulation. And released in the model over a 7-day period based on the condition of the whale at the time of carcass discovery. And this map is before we run the models. You can see in map A, they have a lot of black dots. And this means the race of strain sperm whales. And we use it as a reference to design the red dot. And the red dot is simulated release point. And this triangle is the location from the carcass. And the result of diet lists the raw data of this study. And you can see they have species, number, percentage, and dorsal mental length, and the mass. And this is a simplified chart. You can see they mainly eat on mollusca, and with small proportion with arthropoda and chordata. And in mollusca, they have the squid and the octopus, and in arthropoda, they have decopa, and chordata, they have pyrosoma and the bony fish. And in two-star, also called squid, they consist of 97% by number. And we list the 4 abundant species in two-star, and they comprise over 65% of the all diet. And they also eat non-food items like plastic debris. For example, they are drinking cups and food wrappers. And after we examine this trash, the B and C are originated from Indonesia. And the result of a haplotype. The whale's haplotype is among the most common haplotypes worldwide. But this haplotype appears in the highest frequency in the northern Pacific, especially of Japanese coastal areas, and of Cocos Island in the southern Indian Ocean. And the Caucasus is in the square one. And the modeling, you can see the picture, the triangle is the location from the carcass, and the dots is the drug starting location. And the lines, it means their tracks over 7 days. So we choose the likely tracks and are around the location. So we got the 4 lines. And the discussion, the majority of cephalopods are distributed mesopelagically in the West Pacific, like the 3 species, and with some having a wider or global distribution, like the lower 3 species. So we got the result, and we indicate the whale was frozen outside these relative shallow waters, such as Singapore Strait or surrounding enclosed sea like Java Sea or South China Sea. And what do they eat? Pyrosoma atlanticum. In our study, sperm whales are known to forage at depths in the aphotic mesopelagic zone reaching echolocation. But in this paper shows cephalopods species possessing luminous organs with up to 77.5%. So we suggest it's probable that sperm whales detect and capture food using a combination of echolocation and vision. And because pyrosoma occupies the mesopelagic zone as same as squid, and it's possible that these tunicates are tracked visually in the same way.
Press my icon again. Upper right. Right, you press again.
And the coastal hydrodynamic model, you can see the all tracks are eastward. So the results suggest this whale was likely to be in the West Singapore before she died. And these selected drugs are close to the shipping routes, indicates that she might have a collision with a ship. So in summary, we have the diet evidence and the origin of plastic debris and the hydrodynamic modeling suggest that the sperm whale could have originated from India Ocean, close to the Cocos Islands. However, we could not confidently determine the origin of the Singapore specimen until more detailed genetic sampling and kinship analysis of the sperm whale of Southeast Asia waters can be done. So in conclusion, we know the 3 points about the sperm whale found in Singapore. First, she feeds mainly on small to medium-sized mesopelagic Indo-Western species squid with a smaller proportion of other marine invertebrates and fish. Second, she has the most widespread and common mitochondrial DNA country-region haplotype. Third, results suggest that he could originate from a population in the Indian Ocean. In this study, we provide an increase in our understanding the diet and natural history of the sperm whale in Southeast Asia. And the combined analysis provides a context to future studies on the sperm whale and have broader application on their marine on other marine mammals in the region. And thanks for your attention.
In the diet analysis, what is Physeter means?
His question is, what is two-star mean? Two-star is a group of animal and like Perosoma. So in Chinese is 背囊动物.
Could you please explain the haplotype again?
What's her question?
His question is, what's
Her question.
her question is, what's haplotype? Haplotype can identify patterns of genetic variation. So, for example, if haplotype A is a whale live in Atlantic, so we match this haplotype, we can know this same group. So, it lives in Atlantic too.
I want to know why do you want to choose I forget the whale's name. This kind of whale.
Sperm whale.
What's his question?
Why do I choose the sperm whale?
Right.
The whale specimen is scarce, so if there is a trained whale on land, we can first collect the data for the future studies.
Could you explain what is morphological identification?
In what page?
Method.
You want to know what's morphological identification. So you see with your eye, see the species, and with its characteristic to identify it was the which species. This is
that
answer
clear?
your question.
So I just came in, so I didn't hear how do you like collect the content in the stomach? Do you kill the whale or you just?
Yes, we killed it well.
Oh, really?
So it is dead on land, so we just dissect it.
Oh, okay.
Could you explain why will the sperm whale die on the beach? Is it because either toxic and sick or collide with a ship?
Let me see. Have a collision with a ship, so he died on the ocean, but the floor forced it to the land.
In your discussion, you say it may be collected by the sheep. And does it have any external injury? Or you can say it may be collected by the sheep.
Oh, okay. And in the picture is the news picture and have a large gash on the back. So they have the external injury. So we can say possibly caused by a sheep.
Can you go back to mtDNA?
Yes, this page.
Why choose skin and skeletal muscle to collect?
Because their DNA in skin or skeletal muscle are more abundant. So if we amplify this sequence, it's more probable we can get the sequence.
So I think Jason gave a very nice talk. So he had a very, I won't go back to the slides because it's a little bit wasting time. He had a very clear purpose slide. And you can see that in many of his slides, he added a lot of images. And some of these images are not just for artwork. Some of them are quite informative. Well, I still need to go to. For example, I remember in one of his discussion slides, he talked about where the prey of the whale might have come from. And he showed a very nice map here. Here he said, oh, this might be the location of this mesopelagic prey. I think this is very helpful. Instead of just saying this sea or that sea. Probably some of you might not know where Java Sea is. So I like this slide a lot. And he also had a very specific, I think that's something many of you can learn from his talk. He had a very specific, clear conclusion, which is quite specific. Not just like in the future we want to do this and that. That won't make a good conclusion. Conclusion should be short iteration of what has been found in this study. So far, these are all the good things I saw in his talk. And still, there are something to be improved. For example, there are some difficult terms. I won't go back to these terms, but I can name some of them, like supermacity. What's that? Spermacity. What's that? I have no idea. I'm not a biologist. So I have no idea what that is. And what is drugs? No, you don't explain it now. You explain in your talk too. That's something I just want to point out. You have to think what might be new or unfamiliar for the audience, and then you explain automatically, ideally with the image. And then you can see from the Q&A session that many people are confused. Why do you study mitochondrial haplotype, right? And it is not until your the Q&A session that I have a better understanding why you or the authors chose mitochondrial haplotype. Because this is used to divide different populations of sperm whales. That might be not that easy to do with chromosomal DNA, because that was my question. Why don't they just use chromosomal DNA? Why do they want to use mitochondrial haplotype, which is completely new to me? So next time, please just explain in easier words. I think the slides where you made for mitochondrial DNA did not really fully express the meaning and why they want to use that. So you can improve that next time. And there's a pronunciation benthic, and he printed the table himself. And I think I like his slides of diet analysis where he has this different classification, different layer with many images. I like that a lot. But next time, I suggest that you can talk a little bit slower. especially if there is some terminology in your sentence, then you should always speak slowlier and stop whenever there's a term which you think might be difficult for the audience, because they are not specialists, so that's why you need to explain. And if possible, try to explain in easier words, because some of them I still have the feeling that they might have their original form in the paper, but the sentence in the paper are not actually easy to express to the public. But overall, I think it's a very good talk, so let's thank Jason.